So good afternoon, everybody, and welcome to what I'm going to call a celebration, a new phytologist family celebration. And it's a celebration because it's an enormous pleasure to be able to introduce a lecture by Francis Martin to us today. Before I do that, and before I give Francis a short introduction, I want to extend a really very special welcome to the new phytologist advisors who are present. I think it's an enormous pity for me that I'm not able to meet you in person, because really what I want to do, and on behalf of everybody at New Phytologist, I really do want to, first of all, extend a welcome to you, but also a real heartfelt thanks to each and every one of you, because your involvement with the journal is so important to us. And I hope that at a future meeting, when we meet face to face, I'll be able to say thanks so much in person, and we'll get a chance to know each other better, and you will get a chance to meet other people in what I call the New Phytologist family. Having said that, let's now get back to the reason we're all here. Now, I thought when I was putting this together, thinking about what introductory remarks I would make about Francis, I thought a short biography would be useful and important and interesting. And then, of course, I was reminded of the fact that Francis has published a simply lovely profile. And perhaps, Sarah, you could put a link to the profile in the chat. For those of you who haven't read it, it's beautiful. It describes his motivation and real passion for science. And I think that there's very little more to be said. It's a beautiful piece. What I will say, however, is that I think we're very privileged today. I regard, and the international community regards, Francis as an absolutely outstanding scientist. He is really one of the mycorrhizal scientists of his generation. So it's a great privilege to hear from him today. He's also been an editor of New Phytologist for, frankly, quite a long time. In fact, even longer than I have. And he's been a tremendous supporter and friend to the journal. On top of that, I would add, and you all know this, he is a simply wonderful human being. And it is a pleasure to interact with him, and has been ever since the day I first met him. Now, I'm not the only person to think of this, and I don't want to run through Francis's sort of CV and embarrass him. But when I say I'm not the only person to recognize this, I'm very pleased to say the government of France have recognized this too. And of course, as many of you will know, he has been awarded the highest possible honor in France, which is the Legion of Honor. And of course, he greatly deserves this, and we celebrate this with him. So having made these introductory remarks, what Francis will be doing is he'll be giving a lecture, and very graciously, he has decided that he'd be very willing to answer questions at the end. So we'll have perhaps 10, 15 minutes of questions. Having made these brief introductory remarks, Francis, I think it's over to you. Thank you very much.
Thank you very much. I mean, I'm very pleased to have been invited to give this lecture. You know, as you emphasized, the New Phytologist has been part of my family for many, many years. I'm currently probably the oldest New Phyt editor. And so I'm very proud to be part of the family. And I'm very proud today to talk on before such a very prestigious audience, the editors of New Phytologist, the advisors. It's a large group of outstanding scientists. So today I have the opportunity to summarize in half an hour, 40 minutes, what we have done over the last 5 years, aiming to bridge ecology and genomics and molecular biology. you know that that's one of the main goals of new phytology, bridging the different fields of research. So I'm going to show you how we have used large-scale genomics to get a better understanding of the interactions between trees and a group of fungi I love, the ectomycorrhizal fungi. So let's walk along this path and in the woods. So I'm going to split my talk in four parts, four sections. A brief introduction on the main goals of this research program, the plant microbe, understanding the plant microbe interface in forest ecosystems. Then I will focus on our aiming to relate genome features to ecological traits. That's really the core of our research activities currently, focusing on one of the major traits in forest ecosystem, the lignocellulose decomposition machinery. And then I will show you how along the evolution of these fungi, they gain genes to talk to the trees. And at the end, I will try to wrap up by providing some takeaway points. So this research is carried out in collaboration with many peoples. So today I'm talking on behalf of the Mycorrhizal Genomics Initiative Consortiums, but also a collaboration with our colleagues in the Oak Ridge National Lab in the US. And we are all working within the framework of a project called the Plant Microbe Interfaces. And in this project, the main goal is to characterize the microbial interfaces which allow the transfer of energy, so mainly carbon, information and nutrients between, on one side, trees and ectomycorrhizal fungi, symbiotic fungi. Of course, as many groups today, we worry about the impact of climate change on forest ecosystems. So this program is focusing mostly on the carbon cycling and carbon sequestration. And so I would say we have split this large-scale projects on the interface between fungi and trees in four main objectives. We want to describe and define the community of trees and the community of fungi interacting with these trees. We want to characterize the molecular mechanisms and biochemical mechanisms taking place at these interfaces between fungi and trees. We are aiming also to manipulate a few of these systems. We are lucky enough to have model systems where we can manipulate gene expression. So we want to manipulate these genes to see how they impact the interactions between the two partners. And we collect all this information to try to predict the fluxes of carbon and nutrients in forest ecosystems to feed predictive models. Again, trying to understand the impact of climate change, environmental changes on the forest ecosystems. So I'm sure you know that in woods, there are at least three main types of fungi. And we are trying to use large-scale, to harness large-scale genomics to understand the role of these fungi, focusing again on the ectomycorrhizal fungi in forest ecosystems. But in fact, we know very little about the evolutionary and biological, ecological interactions between these different groups of fungi. So when you go to a forest, a temperate forest or a boreal forest, you have these guys on the left side of the slide. You have the saprotrophic species, those able to use organic matter to decay, to decompose organic matter to feed their metabolism. So you may recognize on the top left of the slide the white rotors, like Trametes, and these fungi are able to decompose, to decay, both lignin and cellulose, and to mineralize them in simple sugars. You have the brown rotors, again, able to decompose wood, but they do it mostly, at least for regarding the lignin decomposition, by using the Fenton reaction, which is a oxidoreductase chemical activity. And then one group of fungi at the bottom left of the slide, the litter soil decayers, play a key role because these guys are really the one involved in the leaf decaying and are able to decompose twigs and branches. So these three groups of fungi, the white rotors, the brown rotors, and the litter and soil decayers, play a key role in the forest because they are the master of decomposition. They recycle carbon. On the right side of the slide, you have the mycorrhizal symbionts, including ectomycorrhizal fungi like the fly agaric that you see there, and arbuscular mycorrhizae. These fungi are able to dialogue with the trees and to set up a mutualistic symbiosis where the fungi are fed, are able to use the simple carbohydrates from the plants, and conversely, they can provide soil minerals and water to the plant. So it's really a mutualistic symbiosis where the two partners are gaining from the interactions. And at the bottom right of the slide, you have the pathogenic species, which are killing their host to feed them. So we know that all these fungi, guiles of fungi, the saprotrophic species, the symbiotic fungi, and the pathogenic ones are key players in forest ecosystem. But we know very little about their interactions and how they evolved during the evolution of fungi. Getting back to the ectomycorrhizal fungi, I'm sure all people working for new phytologists knows about these ectomycorrhizal fungi, but I'm just going to summarize what we know about the biological role and ecological role of these guiles of fungi. We know that ectomycorrhizal, you should know, I think, that ectomycorrhizal fungi are symbiotic species. As I said, they play a crucial role in nutrient acquisition in most forest ecosystems. We know that this ectomycorrhizal symbiosis evolved at least 60 times during the evolution of fungi in Ascomycota, in Basidiomycota, and in some Mucoromycotina. Undoubtedly, the ectomycorrhizal fungi play a key role in carbon cycling in forest ecosystems, but as Bjorn Lindahl would say, there's a considerable variation in their capacity to acquire carbon from soil organic matter. And I will get back to this key point later. So there's currently a debate in our mycorrhizal community about the role of ectomycorrhizal fungi in the decomposition of soil organic matter. Most people think that these fungi can decompose organic matter, but without using the released carbohydrates. So they are not saprotroph. And Bjorn Lindahl summarized this concept a few years ago in one of the Tansley Insights, which has been highly cited more than 400 times, where Bjorn and Anders Tunlid summarize what we know today about the role of the saprotrophs and mycorrhizal fungi in carbon and other nutrient cycling in forest ecosystems. So on this cartoon, you see that saprotroph in blue, clearly they are able to hydrolyze, oxidize, mineralize the soil organic matter. So that's what we call decomposition. But regarding mycorrhizal fungi in red, we know that they are using mostly plant carbohydrates, but some people, some groups, some teams think that they maybe also be able to use partly soil organic matter. So we have been using genomics to try to tackle this question, whether mycorrhizal fungi are or are not able to decay substantially soil organic matter. So we have two main aims in this project: relating genome features to key ecological traits, and I'm going to focus mainly on soil organic matter decomposition. And then we have also tried to track the multiple independent lifestyle transitions from saprotrophism to mutualism in these fungal lineages. And so to do that, we have used, on the left side of the slide, you will see the microcosm database. So we have been collaborating with the JGI, the Joint Genome Institute, to sequence hundreds of saprotrophic species and symbiotic species all along the tree of life of fungi. And today we have sequenced more than 2,000 genomes, and they are available in the microcosm database. We have selected these genomes based on phylogenetic features, but also we have chosen species which are ecologically relevant. So we have a fairly large set of species and genomes. And thanks to a collaboration with many people, and you see there on a few faces you may know, Bernard and Rissa, an expert in carbohydrate active enzymes, Igor Grigoriev, who is leading the fungal genome program at the JGI, David Hibbett, who is a very great, outstanding expert in phylogenetics, and Laszlo Nage in phylogenomics. So two of our younger colleagues, Shingo Miyoshi and Eniko Kiss, we have compared the genomes of 135 species, including 62 mycorrhizal species and 73 saprotrophic, endophytic, and pathogenic species. So it's a really large set of genomes that we have compared there. And there are a lot of benefits of having such a large set of fungal genomes associated to plants. This large set of genomes allowed us to sample major fungal families for which there were previously no symbiotic genomes available. This includes Russula, Teleflora, Cantarella species. And these Russula and Teleflora-less species are really ecologically dominant in most temperate and boreal forest. The chanterelles, like the golden chanterelles you see there, they are very interesting because they represent early divergent clades of symbiotic species. And again, there were no genomes available before that study. And we included groups that, like the chanterelles, which arose before or after the origin of the lignolytic peroxidase, the enzymes involved in the lignin decomposition, lignin decay. So using these genomes, we have, so you have the tree of life of fungi on the right, the long list of genomes we have sequenced. Then we annotated these genomes. So we have now gene repertoires, and we have compared the functional divergence between these species, focusing on gene acquisition, gene decay, gene duplication, gene neofunctionalization, gene family expansion, and gene family contractions, aiming to link gene content to ecological traits. And these traits, again, are mainly the organic matter decomposition, but also I will show you some data on nitrogen and phosphate uptake. So acquisition of carbohydrates, of carbon, acquisition of nitrogen, acquisition of phosphate are three key ecological traits in forest ecosystems. And so we compare these general repertoires for the saprophytic species and the endophytic and symbolic ones. And before providing you with the results, let me remind you the mechanisms, the biochemical mechanisms involved in lignin and cellulose degradation, in the degradation of the polysaccharide, making the biopolymers, making the plant cell walls. So you probably know that cellulose is decomposed by a series of enzymes, so cellulases, exoglucanases, like the GH6 and GH7 enzyme, but also endoglucanases like the GH5 family, GH meaning glycosyl hydrolases. And recently, recently 5 years ago, about 5 years ago, a new family of enzymes have been discovered. The copper monooxygenase is the lytic polysaccharide monooxygenase. So all these guys, all these enzymes are involved in the complex decomposition of cellulose to give rise to simple carbohydrates. And then fungi, some fungi, mainly Basidiomycota, are also able to decompose lignin thanks to a series of secreted enzymes. So you see there the fungal hyphae, the tip of hyphae from a white rot, and this fungus, this mycelium, is able to release a series of peroxidases like manganese peroxidase, lignin peroxidase, lacase, and these enzymes, through a series of oxidoreduction activities, reactions, are able to split, to decay, to cut the lignin biopolymer. So all these enzymes acting on cellulose, hemicellulose, pectins, but also lignin are so-called carbohydrate active enzymes. So in collaboration with Bernard and Rissa, who is a curator of the Casome database, we've been able to scan these genomes and to set up the gene repertoires for these K-enzymes, carbohydrate active enzymes, for all these genomes. So what did we get? So this slide is really a key slide summarizing what we have obtained by comparing these genomes and focusing on some of the carbohydrate active enzymes, nine genes coding for enzymes which are key players in the decomposition of lignin and cellulose. So you have POD, peroxidase of class 2, so they are called lignin peroxidase. I just mentioned them. You have a series of multi-copper oxidases like the lacases, so they belong to the AA auxiliary enzymes. We have the LPMOs, AA9, so the lytic cellulose monooxygenase. We have the GH6 cellulase, GH7 cellulase, and some carbohydrate esterase. And many of these enzymes, they use the CBM1 carbohydrate binding module to bind to the cellulose. So any species having genes coding for these 9 enzymes and this CBM1 motif will be able to decompose lignin and cellulose properly. So we counted the number of genes coding for these crucial enzymes in 145 genomes. So you have the list of genomes at the bottom of the slide, the x-axis, and you have the number of genes from 1 to more than 170 secreted enzymes. So those released by the fungi, by the mycelium in the medium. And you see that there is a striking difference in the number of genes coding for these enzymes along these genomes, along these species. On the left part of the graph, you have fungi like Galarina, Coprinus, Macrolepiota, Trametes. They are either white rotors or a soil saprotroph. And these guys have a very large set of enzymes between 170 and 100. Instead, on the right side of the slide, you have genes, you have species with a very low content in genes. So there you see mostly ectomycorrhizal species like Lacaria, Amanita, Swillus, Boletus, Phytologist, and a few brown rot fungi. So clearly, ectomycorrhizal taxa are depleted in peroxidase, cellulase, and many of the enzymes needed for lignocellulose decomposition. So that's really a very clear link between the gene repertoires for K-enzymes and the ecological and the ecological niche of these species. So the whitewaters and the little decomposers are very rich in enzymes. Instead, the ectomycorrhizal fungi are mostly depleted in these enzymes. So on this slide, it's about the same information. There you have on the right side of the slide, the number of genes coding for enzymes acting on cellulose and acellulose, lignin, pod of class 2, and pectins. And you have the list of species here, mostly Basidiomycota. And you see that the number of genes, as I said, is strikingly different between the different GALs. In blue, most of the cells where you have a blue color means a low content in genes. Most of these species are ectomycorrhizal ones. For example, in the middle of the slide, you have the Bacillus, Xerocomus, or the bolids there, the Swillus, are really having a low repertoire of genes. Instead, the soil saprotroph or the wood decayers like Marasmus or Gymnopus or Armillaria have a high load in these genes. And based on these contents in the current, in the genomes of the current species, Laszlo and Nage have been able to reconstruct the gene content of ancestors by using a software called Compare, and it showed clearly that the ancestors of all these species, including the ancestors of the ectomycorrhizal fungi, were high content in genes coding for lignocellulose degradation. So we have showed there that during the evolution of the ectomycorrhizal fungi, which are deriving from saprotrophic ancestors, during the evolution, they have lost most of the enzyme able to act on cellulose and lignin biopolymers. So we did the same kind of surveys focusing on nitrogen acquisition and phosphate acquisition. So there you have the number on this graph. You have the number of genes coding for proteases and phosphatases in the different guiles of fungi, ectomycorrhizae, saprotroph, pathogen, or kill mycorrhizae, wood decayer on the left, the number of genes coding for proteases, phosphatases, and again the K-enzymes. And you see there that there is almost no statistically significant differences between the ectomycorrhizal species and the other forest fungi. So in contrast to carbon acquisition, during the evolution of these ectomycorrhizal symbionts, we don't see any striking difference in the gene contents used for nitrogen and phosphate acquisitions. So if I summarize what I said so far on the process giving rise to the ectomycorrhizal symbiosis, I showed you that the adaptation to a new niche, which is the host boot, a massive gene loss took place. This means that the loss of these genes coding for cellulose and lignin, or plant cell wall degrading enzyme, as we call them, in most ectomycorrhizal fungi, this means that they have lost their saprotrophic ability. This means that this fungi relies fully on the host, on the tree for the carbohydrates load. And in the meantime, this also means that these fungi, when they penetrate in the roots, since they are not able to decay, to degrade plant cell walls, they don't release oligosaccharide elicitors. So we think that part of this mechanism, evolutionary mechanisms, is a way to avoid the plant defense reactions. And we have observed this loss of genes in Basidiomycota, in Ascomycota lineages. So this means that during almost 180 million years ago, since the rise of the ectomycorrhizal symbiosis, in all lineages of fungi, Basidiomycota and Ascomycota lineages, the same mechanism took place. So it's really a very nice example of convergent evolution. But I should say that we also found a few ectomycorrhizal fungi which have kept a substantial repertoire of plant cell wall degrading enzymes. To name them, Gautieria, Morchiliformis, Cortinarius species, Acephala macrosclerotiums are species which have kept a substantial number of genes for these plant cell wall degrading enzymes. But we have showed that the expression of these genes is turned down during the symbiosis. So they still have the repertoire to decay lignin and cellulose, to decay plant cell walls, but they cannot express them during the interaction. So this shows that evolution of the symbiosis took place by gene loss. But then we investigated the conservation of the genes regulated by symbiosis. So this is a complex slide, but let me go through that slide carefully. This is, by transcriptome analysis, we have been able to identify about 6,000 genes which are induced by the mycorrhizae formation, by ectomycorrhizae formation, in about 12 different interactions. Poplar interacting with Laccaria, Phytologist interacting with Eucalyptus, Amanita interacting with Poplar. So altogether, we've collected all the genes induced by symbiosis. This is about 6,000 genes. And we wondered what is the conservation level of these genes among the Basidiomycota and Ascomycota. So we compare, we assess the sequence similarity throughout the tree of life of fungi. So we use BLAST-P as a way to estimate the conservation of these genes, of these protein sequences. So on this heat map, each line represents one of the 6,000 genes induced by symbiosis during mycorrhizal formation. And the sequence of this gene is compared to the gene repertoire, to the genome of 135 fungal species, and you have you cannot read it, but believe me, you have the name of the species at the bottom. So when the gene sequence is highly conserved, according to BLAST-P, so let's say 100 to 80% sequence identity, you get a red. When the sequence conservation is moderate, you have between 40 and 80, it's yellow or orange. And when the sequence conservation is low, it's either pale yellow or even for some of them, no sequence similarity is white. And so when you look at the heat map, you see that about 2/3 of the genes induced by symbiosis have a sequence orthologs in other fungi, including saprotrophic species. So this means that a large part, about 2/3 of the genes used by ectomycorrhizal fungi to differentiate symbiosis are conserved. They are conserved in saprophytic species, and they have been co-opted for the mycorrhizae formation. They are induced in the symbiosis. And these genes are mainly coding for enzymes of the primary metabolism or transporters, membrane transporters, or signaling proteins. And now you also see that about 1/3 of the genes induced by symbiosis are white, or at least providing a white color. And these genes are conserved within a single species. So this is clade-specific or species-specific genes. And these genes are really species-specific, and most of them have no known function. So we focused, only 3 minutes left, so I'm going to go fast for that part of the talk. But in fact, we investigated these genes coding for symbiosis-specific function. And we found that many of these proteins are small secreted proteins. So on this cartoon, you have the hyphae in brown landing on the surface of the plant cells in green at the bottom right of the cartoon. And these small secreted proteins are the red balls. Many of them remain in the interface between the two partners. They are used to glue the hyphae on the surface of the proteins. But a few of them are able to enter the root cells, and there they go quickly to the nuclei of the root cells where they bind to transcriptional factors. And we have showed over the last few years that quite a few of these small secreted proteins induced by mycorrhizae formation, so we call them mycorrhizae-induced small secreted proteins, or MISSPs, like MISSP7, MISSP7.6, or MISSP10, are in fact binding to transcription factors which are involved in the defense reactions. And by interacting with these factors, these small secreted proteins are dampening the plant defense reactions. So this is new genes, species-specific genes. And so this shows that mycorrhizae evolution or ectomycorrhizae evolution also involved the acquisition of new functions and new genes. So to summarize what I told you today, we can say that the transitions from saprotrophy to ectomycorrhizal symbiosis involved at least this series of mechanisms. First conclusion, ancestors of ectomycorrhizal fungi were soil, litter, or wood saprotrophs. To evolution to ectomycorrhizal symbiosis entailed widespread losses of degrading enzymes acting on lignin and cellulose. But again, a considerable variation exists in the capacity of these ectomycorrhizal fungi to acquire carbon from soil organic matter. Some of them have kept a substantial repertoire of K-enzymes. We have shown that co-option of genes present in saprotrophic species is also very important to fulfill new symbiotic functions. We found that the diversification of novel lineage-specific symbiosis-induced genes, like the MISSPs, are being used to control the plant defense reaction and metabolisms. And finally, an exciting conclusion showed that gosh. Oh, sorry for that. Convergent evolution of hundreds. So these mechanisms are took place in hundreds of fungal lineages. So this one thing you should summarize to go to ectomycorrhizal symbiosis, two main mechanisms: adaptation via gene loss and adaptation via gene gains. So now let me thank you, a few people there in my lab on the left. Claire Fouet is in charge of the effector analysis, Annegret transcriptomics, Emmanuel genome comparison. We have been working we've been very lucky to have a nice group of postdoc fellows, Shingo, Brian, Mara, and many PhD students have been involved in the project. My colleagues in China, my group in China, also working on this comparative genomics work. And then on the right, people I already introduced to you who are part of the Microgenome Initiative. So thank you for your attention, and I'm ready to take a few questions.
Okay, thank you very much, Francis. Basically, there are two ways we can take questions. So, people can either raise their hand and we'll identify you and we'll take it from there. Or also you can use the chat function. And Sarah and I and other people in central office will keep an eye on this. So let me open the proceedings now, and Francis would be very happy to take questions. Who'd like to start things?
I think Keith has his hand up first, so.
Okay, thanks so much. That was a great talk, Francis.
Thank you.
One thing that was kind of interesting to me was, and obviously I'm not an expert on this, but you mentioned that some taxa of these ectomycorrhiza or mycorrhiza retain plant cell wall degrading enzymes, and although they're repressed during interaction with the plant, do you know if they are used in particular circumstances? Are these enzymes still used by the fungi?
If I take one group of these fungi, which is very abundant in most of the forest ecosystems, in the boreal and temperate ecosystems, like the Cortinarius. The Cortinarius, according to Bjorn Lindale work, are probably very active in the soil. So they are probably used to what Bjorn called the litter bleaching. These peroxidase, some of the enzymes which have been retained are what we call atypical peroxidases. And these peroxidases are probably involved in that litter bleaching. So they are turned off likely during the interactions within the root tissues, but expressed in the web of hyphae colonizing the litter or the soil. And maybe Bjorn can comment. I see Bjorn is there. He can probably confirm or complete what I said. Bjorn, would you?
I can confirm. We recently got metatranscriptomic data from some forest plots in Sweden, and about 50% of the peroxidase transcripts could be assigned to Cortinarius species, whereas the rest was mainly Mycena and different saprotrophs. So they are highly expressed in some ecosystems.
And we found this. We found that some ascomycetes, like a guy called Acephala microsclerosium, Acephala, is also retaining as a very high load, in fact, very high repertoires of K-enzymes. But when we do sections of the mycorrhizae, we notice that it's a very primitive ectomycorrhizae with almost no mental sheath around the roots and only a very weak articulate within the roots. So I think that they are probably in the way, maybe in 10 million years from now, there will be nice ectomycorrhizal symbionts, but they are probably, they have a dual way of life. They are still able to decay some organic matter, but sometimes they get in the roots where they are learning to be symbionts. I mean, they are in the process to be symbionts.
They're in a kind of an evolutionary transitional period.
Yeah, exactly. So I think that we need more genomes for this, what we call primitive ectomycorrhizae.
Great. Thanks a lot, Francis.
I think Stefan was next with his hand up.
Yeah, thank you very much, Francis. I have two short questions. So the first one, you show that there is a reduction in these cazyme repertoires, and especially peroxidases and cellulases in the genomes of ectomycorrhizal fungi. But I'm wondering whether this is actually the cause or the consequence of the symbiosis. So based on what you just said, I guess it's more the consequences. But I just want to have your opinion on this. And the second question is, then what do you expect if you would ectopically express a cazyme from a saprotroph, for example, encoding cellulase in an ectomycorrhizal fungi? Would you expect a negative effect on the symbiosis through maybe activation of the immune system?
Well, two key questions. I would say my feeling that this gene coding, these genes coding for cellulases or ligninase and the plant cell wall degrading enzymes, they are lost because the plant is providing the carbohydrates. So it's an effect of the symbiosis. Still, some people have showed that in groups in clades like the Russulaceae, some of the saprotrophic species have lost. I mean, some of the ancestors of the ectomycorrhizal Russula, some of the recent ancestors of the ectomycorrhizal Russulas have lost part of this repertoire. So they are talking about a kind of predisposition for some species, maybe have some kind of predisposition for ectomycorrhizae. It's easier for them to lose these enzymes because they are already in the process to losing them. I'm not really convinced about that. We have two examples, one in the Russulaceae and one in the Amanitaceae. For the rest, I think that most of the extent ectomycorrhizal fungi, they have lost these genes because they don't need them. And for example, in the truffles, we have been able to track the decay of these genes. When we compare the black truffle of Perigord, the tuber melanosporum, tuber aestivum, or tuber magnatum, we can see in tuber magnatum, so the white truffle of Alba, we can see remains of the cellulases. So most of the truffle have lost the genes, but still we have found a few species of truffles having remnants of the genes. So this is really, again, a mechanism in action. Mm-hmm. And the second question was about what's going on, what would be the impact of genetic engineering, what would be the impact of transferring a cellulase like a GH6 cellulase into an ectomycorrhizal symbiont, right? That's what the question.
Yeah, whether this would impair the symbiosis or at least reduce the symbiosis efficiency, maybe via activation of the immune system.
Yeah, I think then the plant cell wall of the host will be partly degraded, release oligosaccharides, which may act as elicitors of the plant difference reactions. So, but we've never been able to do the experiments. As you may know, it's very difficult to master genetic engineering in most of these species. So far, until now, we've not been able to take a gene from a cazyme, a cellulase gene from a saprotroph, and transfer it in an ectomycorrhizal species. It should be done, but never been able to do that so far.
Thank you very much.
And Andre was next up with his hand.
Thank you very much, Francis. This was really impressive data set that you presented here, and very stimulating thoughts. I have actually two questions. So one is, when you look at those small secreted proteins that you were talking about towards the end, manipulating the plant's defense responses, this is literally the same mechanism through which pathogenic fungi can overcome the plant, right? And so is there some indication that those are actually the same genes? And I'm going back to, having, rhizobial communities affecting plant metabolism. in a way as if pathogens were present, but they're usually not. So this is kind of, my background going into that. So are they responding in the same way as the pathogens? And if not, or even if, is there, from the plant's perspective, a different, or any indication that there's a different evolutionary trajectory when they interact with a symbiont versus with a pathogen? And my second question is actually, so you very nicely showed, the loss of the cell wall degrading enzymes, and at the same time, the overcoming of the defense responses. And how far do you think this is generalizable? So these two mechanisms, right, is generalizable across all, let's say, symbiotic or maybe mutualistic interactions between plants and other organisms?
Well, regarding the mycorrhizae-induced small secreted proteins, as I mentioned, most of them are species specific, or at least genus specific. So, for example, the ones we have investigated in the most details, those from the fungus called Lacaria bicolor, they are really specific to Lacaria. When you do a blast P or when you carefully curate the genomes, there is no orthodox. So each clade, each lineage of ectomycorrhizal fungi, or most, have small secreted proteins. Most have mycorrhizae-induced small secreted proteins, but there is no sequence similarity. And I think it's Ralph will confirm that it's very similar in pathogenic species. Most of these effector, what we call effector-like proteins, are really species specific, maybe sometimes race specific. But what we can, so this is the same in the ectomycorrhizal fungi we have investigated so far. But what we can confirm is that there are, at the sequence level, there are differences, but we found in several species, small secreted proteins acting the same way and having the same targets. For example, MISSP7 is targeting the jasmonate receptor in poplar and then turn down the defense reaction by interacting with jasmonate receptor. We found the same small secreted proteins in Phytologist interacting with eucalypts. But there's no sequence similarity, but they act the same. So I think that many of these fungi, ectomycorrhizae, pathogenic ones, they learned how to recognize the plant hubs, like the jasmonate receptor. And during the evolution, they evolved different genes, different sequences. Maybe the 3D structure of the proteins are similar, I don't know, but at least all these fungi, and I think nematodes and aphids and all the organisms interacting with the plants, they learned how to control the plants. So that's the first question. The second one, I forgot what you asked.
The second actually goes exactly in that direction. I was hoping you would come to the aphids and things like that that manipulate. Yeah. The question was whether or not the basic principle that you have shown that will allow the plant to actually interact with the fungus in a mutualistic way, whether or not you think this is general across all symbiotic or mutualistic interactions.
My feeling that we tend to categorize organisms. So we have the symbionts, we have the mutualistic, we have the pathogenic. In fact, they don't mind all these fungi, all these microbes. What they want is to eat the plants in some way, and so I think they are using the same mechanisms to control the plants.
Thank you very much.
Thank you, Andre.
We have an awful lot of questions, so hopefully Francis is okay to carry on for another few minutes to try and capture as many as possible. We have a comment from Peter Kennedy in the chat. He says, beautiful talk, Francis. Regarding fungal N and P acquisition, do you have any speculation on why those are generally lower in ECM fungi than in saprotrophic and other guilds? One might think ECM fungi would specialise in N and P acquisition, given their release from need for environmental C acquisition and their need to share N and P with their host.
I'm not sure I understand.
Sorry. So it's in the chat. He says, do you have any speculation on why? So regarding fungal N and P acquisition, do you have any speculation on why those are generally lower in ECM fungi than in saprotrophic and other guilds? One might think ECM fungi would specialise in N and P acquisition, given their release from need for environmental C acquisition and their need to share N and P with their host.
Yeah, this is so. It came to a surprise. We were expecting that these ectomycorrhizal species would have a higher number of genes coding for amino acid transporters, for example, or oligopeptide transporters, because we know that it's one of the main activity of this fungal species to colonize the organic matter and then to scavenge nitrogen efficiently and nitrogen and phosphate. But in fact, no, they do it with the same number of genes than the saprotroph, and we have no explanation for that.
Okay, hopefully that covers Peter's questions. Next we move on to Dave Johnson, who has his hand up.
Hi, Francis. Great talk. Thanks very much.
Hi.
It's a similar question to Keith's first one, I guess, but maybe a more extreme example. So the situation with ericoid mycorrhizal fungi, which seem to have so many plant wall degrading enzymes. So are you suggesting that they are operating saprotrophically largely, or are they deriving their carbon from plants mostly, or what? So how confident are you that that expression of those enzymes reflects a sort of true function?
Yeah, so good point. I had no time to go in detail through the slide, but you have sharp eyes, and you've seen that many of the ericoid fungi, I would say all ericoid fungi we have sequenced so far, and all orchid fungi we have sequenced so far, have the highest content in genes coding for this carbohydrate active enzyme. So they have a very high potential of plant cell wall and organic matter degradation. But keep in mind, for the people who are not mycorrhizists, we have to keep in mind that most of these fungi are in fact providing, at least the orchid mycorrhizal fungi, are providing carbon to the plants. Regarding the ericoid one, I think they are mainly saprotrophic species living on sphagnum mosses, and then sometimes they do interact with plants like vaccinium. And in that case, most of the enzymes they are encoding are again turned off, but not all, because in ericoid mycorrhizae, the fungal hyphae should penetrate within the air root cells. So they are using these enzymes to penetrate within the cell and to make what we call the peloton within the cell. In ectomycorrhizae, you know that, Dave, but all the people should know that the ectomycorrhizal fungi, the hyphae, remain outside the cells, so they don't need enzymes to penetrate within the root cells. So I think the ericoid fungi are probably have a dual mode of nutrition. Most of the time they use the sphagnum moss as a substrate, and sometimes they do interact with the plants. And that's one point. And the last one is the fact that they are considered as young symbiosis, among the younger symbiosis which evolved only 100 million years. So this also could be getting back to what we talked with Keith, it's maybe a system in transition. Would you agree with that comment, Bjorn and Dave? Or?
Yeah, that sounds convincing to me. That's, yeah.
Okay. Thanks.
Yeah, to me too.
Perhaps we can take Jennifer's talk. Sorry, Jennifer's question next.
Thank you for a really wonderful talk. I see that in your there's a high number of high gene gain in the Permian after the Carboniferous, and I wondered, could you use your comparative framework to look at the hypothesis of the origin of lignin degradation for the Carboniferous particularly?
Yeah, we have, that's mainly the work from Laszlo Nagy and Dimitri Floudas in David Ibet's group, where they showed that they've been able to trace back the emergence of the lignin pod, the class 2 peroxidase, at about 280 million years ago. And when these fungi, these ancestors of fungi were able to, thanks to a mutation, then this class 2 peroxidase evolved, and then they've been able to use lignin. And that's probably why they have been able to radiate so massively. I mean, it was such a benefit to get access to wood that this has been a tremendous successful step in the evolution of fungi, or at least the Basidiomycota. And from these radiations, much later, probably around 100 million years ago, ectomycorrhizal fungi evolved. So this is really a multi-step process for the Basidiomycota. Invention of the class 2 peroxidase to allow the decomposition of wood, then a massive radiation of Basidiomycota. And within some of these families of Basidiomycota, step by step, brown rotors evolved, and then much later, the ectomycorrhizal fungi. Does it fit your ideas?
Yeah, so it does. You suggest then that a lack of lignin degradation in the Carboniferous then could have contributed, let's say, to high oxygen in the atmosphere if you don't have that degradation. So that's the kind of, it's really interesting to see.
Yeah.
So Carlos' hand has been up for a while, but I'm not sure if this question was covered in Keith's question. So if it's a separate one.
Okay, I guess my question is related to some of the point that Andrea raised about comparisons with pathogens. I think this was a brilliant talk, Martin, Francis. But I wonder your talk implies that the main reason why ECM lost plant cell wall degrading enzymes is to avoid the generation of oligosaccharides and triggering plant defenses. And that would also be an advantage for a biotrophic pathogen. And so I wonder if you see a similar trend for the pathogens that the pathogen genomes that you sequenced, this reduction in the number of genes encoding for plant cell wall degradation.
Good question.
And also
yeah.
for a necrotroph, which like botrytis, which could be a saprophyte also. I wonder if these genes are expressed as a function of context, basically.
Okay, I'm not sure I understood the second question.
Well, I wonder what happened with the necrotrophs, which like botrytis, which could be.
What we found we also sequenced many rust fungi, so biotrophic species. And they behave very, the genome features of these rusts is very similar to the ones from ectomycorrhizal fungi. They are able to penetrate, they have kept pectinases to penetrate within the cells. They have kept a few endoglucanases, but then they have lost many of these enzymes to probably stealth in the leaf. So yes, rust biotrophic fungi have a very similar genomic features to ectomycorrhizal species. Instead, the necrotrophic species, then they have kept a huge number of plant cell wall degrading enzymes. So there is, again, a striking difference between the necrotrophic species and the biotrophic species.
Thank you.
Okay.
Do hate to draw this absolutely fascinating discussion to something of a close, but we've almost given Francis 25 minutes of questions. I wonder if perhaps we've got time for just a couple more. I mean, under normal circumstances, I would stop it here and say, right, let's go and get a coffee, let's go and get a beer and continue this discussion. But we can't do that. So, Sarah, are there a couple of other ones which we should ask?
Yeah, so we've got obviously there's a few people still with their hand up. There's a few more questions in the chat. One from one from Martin. He says, since the roots are also hotspots of bacterial colonization, I was wondering whether there is any evidence for horizontal gene transfer of genes from the bacteria to mycorrhizal fungi. I just remembered the work on bacteria in Lacaria bicolor hyphae.
Horizontal gene transfer, we checked for that, and so far no convincing results. So I don't think that horizontal gene transfer is a mechanism of gene exchanges in ectomycorrhizal fungi, even if they live together very intimately. As far as we can tell, there is no transfer of RNA or genes between the two partners.
Okay, perhaps I can spot somebody. I've seen that in the chat, Christelle. I think Christelle has a question. Would you like to ask it personally, Christelle?
Oh, I'd be happy to. Thanks for the talk. It was really brilliant. I much enjoyed learning so much about ectomycorrhiza. And so I'm not that familiar with it, but I always wonder, if there is a range from mutualistic to pathogenic, and especially in this case where you have this primitive ectomycorrhiza, which can still activate this gene, this lignin degrading genes, whether they could turn pathogenic onto the plants when the plant is so stressed out that they have no resource to give to the fungi anymore.
Okay, so the question is whether we have found any what we can say, whether some of the ectomycorrhizal fungi could turn to pathogens.
Exactly, under stressing conditions for the plants.
So far, never. We never. So you have to keep in mind that we have sequenced only 200 genomes, 200 species, which is huge. But it's only, you should know that there are 20,000 species of ectomycorrhizal fungi. So we have just scratched the surface. And so, but till now, we never found a clade of pathogenic species deriving from an ectomycorrhizal symbionts. In all cases so far, we found ectomycorrhizal symbionts deriving from saprotrophic species. We never found a reversion from ectomycorrhizae to saprotrophy, not yet, and never found ectomycorrhizae to pathogenic species.
All right, thank you.
Okay, thanks very much, everybody. I think we should let Francis off the hook, but I think I can probably say on behalf of everybody here, Francis, that was wonderful.
Thank you very much to you all. I mean, it was a real pleasure to interact with the board this way. So hopefully we can meet again next year and talk about mycorrhizae evolution in a nice restaurant with a nice bottle of wine.
I think that would be a great idea. And I also say, I should say that for somebody who's spent almost his entire career working in signal transduction and stomata, yeah, I'm thinking, is it still too late to get into mycorrhiza? Because it sounds interesting to me. I don't say that very often. Anyway, let me, on your behalf, everybody, Francis, thanks again. That was wonderful.
My pleasure.
It's everything I expected. Needed more.